Hi, everyone. Welcome to the Physionic Podcast. I am live streaming this for the first time in probably a few months. I just haven't had the time to live stream. And considering this week is a little bit more relaxed in the lab and schoolwork wise, I decided to, why not, cover this paper, which could be pretty controversial, actually. If you're unfamiliar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, and I guess my other credential I should mention is that I worked in a metabolism laboratory for a number of years, and I do have some nutrition science training as well. Okay, so this particular paper that I'm going to be going into, or I'm going to be discussing a study, a Nature paper, which is incredibly complex, but we're going to be going over a few different key data points. And essentially what the paper is looking at is the effect that the ketogenic diet has on two things. One, heart fibrosis, so essentially creating fibrosis within the actual heart structure, which is unfavorable. It is a pathology. That's not what you want. And the other thing is to look at mitochondria. So they wanted to look at mitochondrial content. So we're going to be breaking those two down based off of the limited data that I've kind of put together. There's much, much more than what I'm going to be covering in this podcast. And certainly I'll be covering that in future content. But let's get a bit of background out of the way. The ketogenic diet has been used for a variety of different conditions, like epilepsy, weight loss. Obviously, weight loss is a huge one. It's probably the main reason why people use it. It's also highly effective against diabetes, obviously because you're cutting out carbohydrates. You're massively reducing your carbohydrate intake. You're consuming anywhere, it could be less than 10%, 5% of your total energy intake. And some people just don't consume carbohydrates at all, or try not to, among other pathologies that the ketogenic diet helps with. So it's characterized more specifically with a high fat, low protein, and very low, if not non-existent, nutrient profile. So carbohydrate nutrient profile. So ultimately, that leads to your ketone levels, and the reason why I'm mentioning the ketones is because this is going to be important as we go throughout this study, that the ketones are what are produced in ketogenesis by the liver. So the liver will take energy and essentially restructure the molecules to create ketones and release those into the bloodstream. And then those get taken up by other tissues, and those undergo a process called ketolysis, where they end up breaking those ketones down into and back into energy to be used for, the energetic needs of whatever cells end up taking those ketones. So usually normal ketone concentrations are between 100 and 250 micromoles. You don't really have to know what a micromole is. I'm going to explain sort of how to look at it. But when on keto, ketone concentration can rise over 30 times. And that's really the key point that I'm trying to get across here, that kind of in regular situations where you're maybe consuming carbohydrates, you have really low ketone levels in the bloodstream. And then once you end up employing, let's say, a multi-day fast, as we've seen on Physionic from previous content, definitely, but which is actually the most potent way to increase your ketones, but also through a ketogenic diet, you'll slowly see a rise in those ketone levels, and so they can get over 30 times. I think I actually picked a low value for that, so it can get much higher than 30 times the normal carbohydrate consuming levels. And of late, so the last thing I wanted to point out is that of late, the ketogenic diet has been under scrutiny for having potentially negative effects on the heart in clinical trials. So that's been something that this particular group of researchers are trying to get to the bottom of. What could be the reason why people are getting these results? And I should note, however, that, and the researchers also note that they could also be talking about diabetics. So diabetics, you think of diabetes as a high blood sugar situation, which is certainly the case. However, in extremely advanced diabetes, you can have what's called ketoacidosis, where you get the overproduction of ketones as well, because the body or the cells are so insulin resistant that they can't even take up any more glucose, blood sugar. So that elevation in ketones is then associated with a greater risk of stroke and heart disease. Now, something that I should point out is that there is a difference. There's likely a difference between ketoacidosis, which is a result of a pathology like diabetes, and just undergoing ketosis as well. Let's say you change your diet. Obviously, it's going to be a pretty big difference, because you can just stop your diet and then you'll be out of this high ketone situation in your bloodstream. However, while that is important, that may not entirely matter according to the data that I'm going to be presenting in this study. So briefly on the study design, the researchers put rats on 3 different conditions, 3 different diets. And yes, this is in rats, but there's another component to this study. So they put the rats on a normal diet, so the regular chow, calorie restricted diet, so 30% calorie restriction, and the final one was a ketogenic diet. Then the other part of the study is actually looking at humans, where they took heart samples from people that were undergoing heart transplant, and they ended up taking tissues from healthy, or I guess non-fibrotic hearts as well as fibrotic hearts. And fibrosis is essentially just the infiltration of collagen and different structural proteins in the heart. Now, obviously, the heart muscle should be predominantly muscle. You want that to be able to contract effectively. And if you have the infiltration of fibrosis, then that means that you're taking away space from the heart cells that could be there. Okay, so that's kind of the 2-part system. We've got an animal model to actually dive into some of the mechanisms, and then we've got these samples from these human patients. Okay, so let's look at kind of the first bit of data. The first bit of data is literally just, and this is in animals, they're just looking at the diameter of the heart, so looking at the size of the heart. And if you're watching the podcast, not that you're obligated to, if you're just listening, I'm going to throw up a picture here of just kind of a rough idea of what we're looking at here. Essentially, you have a normal heart on the left, and then you have a hypertrophic heart, what I labeled as a hypertrophic heart, on the right. And what they found is that when these animals were put on a ketogenic diet, that they ended up developing a larger heart. Now, a larger heart by itself doesn't necessarily mean that you are going to have any sort of pathology. Now, you can get an enlarged heart from other things as well. For example, if you're a marathon or any sort of endurance athlete, your heart can grow tremendously. However, it's the type of increase in size of the heart and really the areas of the heart that end up growing. So I've pointed arrows to kind of what's known as the septum of the heart, as well as the wall of the heart. So the wall thickness increases so much that the chambers of the heart start to decrease in size. So yes, that would then also be considered a cardiomyopathy. It would be considered a hypertrophic cardiomyopathy. So yes, it is an increase in the size of the heart, which is considered pathological. So that's what they're kind of alluding to here, but that doesn't this isn't definitive proof that this is exactly what's happening. But the ketogenic diet in these animals ended up leading to an increasing in the size of the heart itself. Now, this first piece of data is, I've labeled this, and of course I'll describe it for those of you that are just listening to the podcast. This first piece of data is essentially what's called a microscopy image. So it's microscope images. And this is actually something that I do a lot in the lab. And this is where they take a sample of the heart and then they plate it, and then they they essentially use the microscope, a really high-powered microscope, and are able to see individual cells. And they're not so much interested in the cells themselves, but they are interested in the coloration. And what I mean by that is that they add a particular dye, or they might add a series of different dyes to kind of determine what is what within a particular region of the heart. So they have what's called an H&E stain, a sinus red stain, and a Mason stain. And the Mason stain is the one that we're actually interested in this situation, because by comparison of the normal diet heart to the calorie restricted heart to the ketogenic diet heart, what they find is that anywhere you see blue, that is indicative of collagen. So that's because that dye is specific to collagen. So what it does is it essentially implies that there's greater fibrosis in whatever images you see that, or the researchers see, that have this blue. And what they found is that the normal diet had no fibrosis, the calorie restricted had no fibrosis, but then the ketogenic diet condition did have fibrosis. So this is kind of, again, just pushing into this narrative of, kind of what they're looking at. Another aspect that they did is, which I actually don't have images for, is they ended up doing the exact same thing, but they wanted to look at not the ketogenic diet, but they wanted to specifically figure out which ketone would have this impact. Is it all the ketones, or is it just a particular type of ketone? Because there are three primary types of ketones, really two primary types, but I'll cover all three of them. So one of them is beta hydroxybutyrate, which you'll hear me say many times. The other one is acetoacetate, which is a second one, and both of those are produced by the liver. And then you have acetone that's also produced, but that gets breathed out. So if you ever smell the breath of a person who's deep into ketosis, they have kind of a sweet scent to their breath, and part of that is because they're breathing out some of those ketones. So what they ended up finding is that running that same experiment, but instead of, again, putting these mice on a ketogenic diet, what they ended up finding is that when they injected beta hydroxybutyrate, or they injected acetoacetate, the acetoacetate condition did not lead to any sort of fibrosis. However, the beta hydroxybutyrate injection did lead to fibrosis. So now they're starting to really hone in on this idea of like, okay, well, it's not just the ketogenic diet. We're specifically starting to target a ketone that is of interest. So from here on out, they're going to not just focus on the ketogenic diet, but they're also going to try some experiments with beta hydroxybutyrate to figure out if that is truly the ketone that continuously causes potential problems. And what we're going to find at the very end of the study is kind of a nice, they put a nice bow on top of things by returning back to this point in the study and looking at some of the human patients. So in another experiment that they did is actually, again, looking at the heart size, but instead of putting the mice on a ketogenic diet, again, they did the exact same thing where they injected with beta hydroxybutyrate, and yet again, they found that there was that increase in the heart size that I was talking about earlier. Now, again, this does not necessarily mean that this is a negative pathology. It just means that there's change here. We're going to see, however, of course, with the earlier data, also with the microscopy data, that may not be all that favorable. So we're getting change, and now we're starting to see some of the potentially unfavorable situations as a result of that change. So in the next piece of data, what they wanted to look at was look at cell death in the heart. So looking at cell death, they're using a series of different cell lines. So they're actually looking at they're taking cells and they're plating them onto like a dish, and then from here they're doing a variety of different manipulations. So they have, let me go over the cell lines first, and then we'll go into those manipulations. So they have 5 cell lines. Two of them are animal heart cells, so taken from rats. I believe both of them were from rats. Maybe one of them was from mouse. The another 2 were non-heart cells, which are mouse embryonic fibroblasts, I believe. So they're essentially collagen-producing cells, and the same, I think, is also true for the 3T3 cells. So the point is that they have 2 animal heart cells plated, and they have 2. They're not 2 heart cells. It's not single cells. It's, thousands and thousands of cells, but they're all in this dish. So they have 2 sets, I guess I should say, of animal heart cells, and they have 2 sets of non-animal heart cells. And then, of course, because they want to have a little bit of translation to some later data, they also end up plating human heart cells as well. So they've got 5 conditions in that regard. And then they're going to look at cell death. So what they're doing here is looking at cell death. And they've got their control condition, which is essentially no manipulation. They're just looking at the cells and saying, okay, well, this is what we've got when we do nothing. And then from there, they add TSA. And TSA is an apoptosis inducer. What does that mean? Apoptosis is programmed cell death. So when they add this drug in a high enough concentration, then that leads cells to end up dying. And TSA is trichostatin. So, and then the third condition, as you can likely guess, is that they end up adding beta hydroxybutyrate to these cells as well. So from there, then they measure cell apoptosis levels, or the overall cell death, and they find that the rate of apoptosis, or the rate of cell death, is increased, obviously, with the TSA condition, because, well, that makes sense, right? If you're going to be adding a drug that's supposed to kill the cells, you would hope that it would, well, do its job and kill the cells. So you do see an increase. So they did see an increase in all of the cardiomyocytes, meaning all of the heart cells, all died at a faster rate with the addition of TSA. However, by comparison, when they added the beta hydroxybutyrate, again, they found the same results, that you saw an increased rate of cell death. And what's interesting here is that it was specific to the heart cells and was not found in the fibroblasts. So there may be some sort of mechanism that's specific to the heart cells that is not found in the fibroblasts, which I don't think that they end up pursuing any further, but I would be really interested to really know why that might be the case. Especially what I'm interested in is why the TSA had no effect on the fibroblasts, if it is maybe specific to some sort of system within the cardiomyocytes, the heart cells. So that's another piece of data, essentially. And then another area that they wanted to look at was, okay, well, how are these cells being killed off? How are they undergoing apoptosis? Because there's a number of mechanisms for inducing apoptosis. I'm not going to go over all of them, but essentially you can have cells that tell other cells that they need to undergo cell death. And then you have internal mechanisms that can also induce cell death. Some of them are actually mitochondrial motivated. I don't want to say motivated, but they're started by mitochondria, so mitochondrial dependent apoptosis. And then you have other types of apoptosis where the cell does so much damage to itself that it essentially leads to cell death. And how that ends up manifesting can be a number of different ways, but one of the key ways is by attacking the DNA. So you have a cell, and there's a nucleus in that cell, which is an organelle. It's essentially just a ball-like structure. Within that ball, you have all your genes. They're all nicely packed away, and they're nicely guarded off, and they're separated from the rest of the cell. And that's because it holds all of the blueprints of how to essentially maintain that cell. I mean, if that cell wants to continue to live, it has to constantly access the blueprint to be able to know how to read and produce these different proteins that it needs. So obviously, if you end up destroying everything inside the nucleus, the cell tries to access it and it finds that everything's destroyed. So then it can't continue to produce the proteins that it needs, and eventually it starts to degrade, and eventually it ends up undergoing apoptosis. So they wanted to look at this DNA. So here, they're again using microscopy, so they're again using a microscope, and they're using a fluorescence for intact DNA. So the intact DNA was in blue, and the sheared or fragmented DNA was in green. Now, what could that sheared or fragmented DNA mean? It would mean that the DNA is being cut up. So if it's being cut up, it's not a great sign. The cell is highly likely to end up undergoing cell death as a result, especially if if enough DNA damage occurs. So what they found is when the animals were fed a ketogenic diet compared to a normal diet, I don't think that they actually compared this to the calorie-restricted diet, but they compared it to the normal diet, and the ketogenic diet showed higher fluorescence of the green. So in both conditions, you should see some blue, especially in the normal condition. And then they also had a condition where they injected with ketones, or more specifically with the beta-hydroxybutyrate. So as a result, they found that there was, again, heightened expression of, or not expression, but the heightened fluorescence of this fragmented DNA. So essentially, what's probably happened in this situation is that an enzyme that's inside the nucleus called a DNase, so DNASE, has started to cut up all of the DNA. And essentially, the cell is, not that it's really deciding this stuff, this is a bunch of biochemical processes, but ultimately the cell is essentially packing it in. It's just cutting up all its DNA. There's no way to return, to come back from this, and then eventually it's going to undergo apoptosis or cell death. So the ketogenic diet and ketones in both conditions for these rats led to increases in sheared DNA. So far, I mean, we're getting some pretty good evidence that there's some destructive habits that are happening in the cells, specifically in cardiomyocytes, that's leading to all of this, and this is all ketogenic related. However, I just want to put a break on this real quick and just tell you that this isn't going to be an end-all be-all of avoid the ketogenic diet. As hopefully, if you're familiar with physionic long enough, you know that I like to have a very tempered view. I like to take data as it is. This is one study. I'm going to give you a real kind of overall general understanding of what we should be taking away from this study at the very end of this podcast. Okay, so they've looked at ketogenic diet, hydroxybutyrate, the cutting of the DNA. They've looked at its comparison to an apoptosis inducer. And now we know that it has some sort of effect on fibrosis, and it has some sort of effect on heart cells. But now they also were interested in its effect on mitochondria. So in this situation, it's really simple experiment. They're just plating cells, cardiomyocytes or heart cells in this situation, and then they're adding differing levels of ketones. Which ketone? You can likely guess it's beta-hydroxybutyrate yet again. So they're adding different concentrations. So they have a zero millimolar concentration, which means that there's no ketones. So that's your control. That's what you compare against. And then they've got their 2.5 millimolar, 5 millimolar, and 10 millimolar. So essentially doubling every time. And what they showed is, and they tested this in 3 different cell lines. So they tested this in the human heart cells that they tested earlier, and the 2 animal heart cells that they tested earlier as well. And here again, which was consistent across the board, they found that with increasing levels of ketones, that they had reduced mitochondrial DNA. So let me explain that a little bit. What is mitochondrial DNA? If you're not familiar, actually, nuclear DNA, so what I just described in talking about the last piece of data, that yes, the cells do have DNA or genes that are kept within the nucleus of the cells, and that is a major, I mean, that's where most of our DNA is, that's where most of our genes are. They're located in the nucleus. However, the mitochondria actually also have DNA, and it accounts for about, I think, less than 1% of our total DNA. But the DNA that is in the mitochondria is crucial, because without it, we can't actually produce more mitochondrial proteins. And we need those proteins to actually keep our mitochondria functioning. So a reduction in mitochondrial DNA would then mean that you're getting some sort of inhibition of mitochondria. If that's you're destroying more mitochondria, or if that is producing producing less mitochondria, I should say. So what they're doing is they're comparing the total amount of mitochondrial DNA compared to the total amount of nuclear DNA. So you can imagine that if mitochondrial DNA decreases, then that changes that ratio. And that's what they ended up finding, that with greater levels of beta-hydroxybutyrate, that ketone, then they saw decreases in mitochondrial DNA. So on either side, we don't actually know if that leads to greater mitophagy, so the autophagy of mitochondria, or, which is actually what one of the areas that I do research in, or if that also could be because of reduced biogenesis. And I don't actually remember if they ended up trying to figure out the difference between the two. But the overall point is that there's decreases in the mitochondrial DNA. And they did other tests as well. It wasn't just based off of this, because there are certain arguments that you could make against this particular type of test. They did other tests as well, like I believe TMRM tests and stuff like that, which is looking at the polarization of the mitochondria. And there are a series of other tests as well that you can do. But the point is that with that, then they knew, okay, with increases ketones, then you get decreases in mitochondria. Okay, and then the final piece of, well, not the final piece of data, but the second to last piece of data, but the final one that I actually have some visuals for, is looking at so this is what's called a Western blot, and I realize that if people are looking at this, they might think, like, how am I supposed to even read this? The point is that the smudges, if they're darker, then there's more of that protein. It's really that simple. So they ended up figuring out that maybe this has something to do with this signaling protein called sirtuin, but specifically sirtuin 7, which you may have heard is linked to like NAD. It's related to other metabolic processes. But ultimately, and I'm not going to go too much into sirtuins, one, because I need to educate myself further on them, but ultimately they are a signaling protein. And that signaling protein is implicated in inhibiting mitochondrial biogenesis. So I suppose that might offer some credibility to the idea of reducing biogenesis and not going through mitophagy, but it could easily still be both with our previous data looking at the total amount of mitochondria, or DNA more specifically. So what they found is that when they give these cells, or I believe it was from cells, it could have been from the mice, and, oh no, it is cells. So it's in all three of the different cell types, these cardiomyocytes, what they ended up finding is that when they add these different levels of ketones, so 2.5, 5, and 10 millimolar amounts, that it was a stepwise increase. So it's a dose response. So the more ketones you have, the more sirtuin 7 you have. And the association there, although they don't actually, or at least I'm not going to be showing any of this, the association there is that with increased sirtuin 7, you have inhibition, you have stopping or reduction of mitochondrial biogenesis, meaning you have inhibition of the production of more mitochondria. And that might be the reason why you have this effect on mitochondrial DNA, or what they ended up finding. Okay, so now we have this kind of linked sirtuin 7, and there's a lot more data on that, but that's not really what I wanted to focus on, because it does really get pretty molecular, and I don't want people's eyes to glaze over when I go over that stuff, although I do very much enjoy going into it. Maybe in the future I will go into more molecular stuff. Every time I do, though, people are like, that went way over my head, and yeah, it sucks. It can get pretty complex. Okay, so the final piece of data, all this stuff so far that I've been talking about has been in animals, and it's been in vitro work, meaning that it's been in cells. So clearly that leaves some want, let's just put it that way, some want for some human data. Okay, great, this applies to animals, blah, blah, blah. And certainly you can make, animal research is incredibly important. Cell research is incredibly important, but you do want to have some level of like, hey, is this going to translate? And cancer researchers know this better than anyone, that you can have these things that work really well in mice or rats, or, curing them and whatnot, but then actually translating that to, or I should say curing, I guess I should really say more so like treating the disease and really like stopping it in its tracks or something along those lines. But then like passing that on to humans or trying that in humans, a lot of those human trials don't end up panning out for one reason or another. I mean, our DNA is slightly different, so that could be a factor. There are a number of different reasons. Sometimes studies aren't taking into consideration some of the differences. So anyway, but in this situation, they did take this into some consideration. I still have some critiques of this, but they wanted to look at these patient-derived samples of their hearts, right? So they're taking actual tissue samples from the hearts of individuals who had fibrotic hearts, right? And then they're going to take some pieces of that fibrotic heart, and then they're going to test that and see, what differences are there. And they're comparing that against a non-fibrotic heart, or tissue from non-fibrotic hearts. And in this situation, then, they're first going to look at the amount of beta hydroxybutyrate, right? I mean, why not, right? If this whole thing is revolving around cardiac cell death and fibrosis and the production of collagen, they want to know, okay, well, is there a link there, or are we just wasting our time? And this is all just in animals. And what they ended up finding, again, I'm not throwing up the data, although the study is open access, so you can check it out for yourself. And I'll have my notes that release once the more polished version of this releases. But ultimately what they found is that, yes, there were elevated levels of beta hydroxybutyrate in the fibrotic tissue, but not in the normal heart tissue. By comparison, they also did this to acetoacetate just to make sure that this was specific. And yet again, they found that acetoacetate was the same for the fibrotic versus the normal heart tissue. So this again kind of creates this picture of, we're really isolating this one ketone, really focusing on beta hydroxybutyrate at this point. Now, another thing that they ended up looking at was the sirtuin levels in the fibrotic tissue versus the non-fibrotic tissue. And they found, you can probably guess this at this point, that in the fibrotic tissue, sirtuin levels were higher. So that, again, feeds into this story. And then the final thing that they looked at, they looked at a few other things as well, but the final thing that I'm going to be discussing, because it relates to what we've been talking about so far, is that mitochondrial DNA is reduced in the fibrotic tissue as well. Okay, so I realize that was a lot, but I'd like to kind of put this all together for you into kind of a conclusions and takeaways, and keep in mind that this stuff should be relatively tempered, because a lot of this research is it's in animals, it's in cells. That's really interesting. It's necessary to have, but you have to have the translation to humans. And while I did say that they did translate some of this to humans, the problem is that a lot of that was retrospective, meaning that they took the samples and then they looked at the information or looked at the data from those samples. It's not like they're actually doing a manipulation or, they can't do this, but like putting humans on a ketogenic diet or injecting them with ketones and then taking a piece of their heart. That would be incredible, but unfortunately, or maybe fortunately, the ethics committees would probably have a little bit of an issue with that. So we can't do that. So they did what the best that they possibly could in this situation. All right, so what are the conclusions? What are kind of the takeaways? The ketogenic diet, I've got this nicely written out here because I had to think about it beforehand. The ketogenic diet increases heart cell death and increases fibrosis, and it seems to be specific to beta hydroxybutyrate, but not any of the other ketones in rat and human cells, one human line cell. It also lowers mitochondrial content and could be through sirtuin 7, or at least partly through sirtuin 7. Human tissue with fibrosis shows elevated beta hydroxybutyrate and sirtuin 7 expression, which is what we just went over, as well as potentially reduced mitochondria, if you believe the mitochondrial DNA to nuclear DNA ratio. This offers some cell-based non-clinical data, that's key, non-clinical data, that ketosis can lead to unfavorable outcomes for heart cells, and retrospective indirect data, and that's really key there as well, indirect data that ketones are associated with fibrosis, because again, they're taking the sample and then they're testing. So they don't know. It's not like they're adding the ketones to it and making that assessment. This does not prove the ketogenic diet is harmful by itself and is likely more translational to a pathological condition like diabetes, which is what I talked about in the introduction, where ketoacidosis is possible. So the elevation in ketones that's not favorable, that's in a highly pathological body state. If this translates to either clinical situation, regular ketosis, or ketoacidosis, needs actual prospective randomized controls to confirm. So meaning that we would have, just like I described, you would need people that are on a ketogenic diet and then take heart tissue from them. And I suppose that might be possible by doing something like putting people on, or people that are on a ketogenic diet for their entire lives and then decide to donate, their heart to science, then you can do some stuff with that, but it's still not exactly as well controlled as you might like. So overall, this means that this is really intriguing, potentially alarming data, because if this ends up translating, but not to be taken as definitive evidence against any ketogenic diet. So that's where I'm going to leave things. Hopefully you got something out of it. It's really interesting data. Obviously, it's something that would need to be corroborated by most likely other researchers as well. But it is in a extremely high impact journal, Nature. And a ton of researchers worked on this study, and it's certainly something that raises questions, but just one study doesn't necessarily mean that that's going to pan out in the entire literature. So, and as we know, what we definitely know from clinical work is that the ketogenic diet has had many positive effects, in weight loss, and has had an impact in diabetes, specifically in those two areas, but certainly others as well. So, we have to take the data and we can't just ignore it. We have to accept it, but we have to look at it from a critical lens as much as possible and see if some of this stuff can fit in with other data that's out there or other data that will come out. Okay, well, thanks for stopping by, and I wish you a wonderful day, and I'll catch you in the next one, hopefully. See ya.